Hi everyone and welcome back. So in the previous class, we were talking about this dynamic light scattering technique with regard to particle size analysis, and we have seen the basic principle of this technique, and we have discussed as to how the data is collected and how it is analyzed. So the basic principle of this technique is based upon the Brownian motion of particles in a suspension. And due to this Brownian motion, when the particles come in the path of the light, the scattered intensity which is collected on the detector varies. And this variation in the intensity is related to the size of the particles. Okay. So we have seen that this motion can be defined by a parameter called translational diffusion coefficient, which is related to the particle size d by the Stokes-Einstein equation. And if we could derive that parameter, we will be able to get the particle size from this particular equation. Okay. So that is what the dynamic light scattering technique does in terms of measuring those intensity fluctuations which contain this time scale information of this motion, and from that the diffusional coefficient can be obtained and it can be used in this Stokes-Einstein equation, and the particle size can be derived from that. But in order to do that, the data has to be analyzed, and for that we have seen there is a method called autocorrelation which is used to derive this diffusional coefficient from the intensity fluctuation data. Okay. So once you do this autocorrelation with the help of a second order autocorrelation function, which basically correlates the intensity of a signal with respect to the time, you can derive the diffusional coefficient, and from that the particle size can be obtained. Okay. So now in this class we are going to see how the instrumentation is and how the experiment is actually carried out in the DLS instrument to measure the particle size and size distribution. Alright, so here is a schematic of the instrument where you can see the vital parts. The instrument uses a laser as a monochromatic light source, as you can see over here. And apart from that, it consists of a sample cell which is here. The sample cell is placed in the gap between the laser and a detector. The detector is a photodiode array detector which captures the scattered intensity. So a fluid stream with dispersed particles is passed through this sample cell, and as the particles come in front of the light beam, it gets scattered and the scattered intensity is captured by this detector. Okay. Now you can also see there are lenses which help in focusing the scattered intensity onto the detector. So the signal is collected by the detector and sent to the computer where it is being analyzed with the help of that autocorrelation function that we talked about before, and the particle size is derived from that data. Okay. Now we can also see that there are more than one detector over here, like here, and there also. So if you have more than one detector in a system, it allows more flexibility and also helps expand the dynamic ratio which is the ratio of largest to smallest particle size, which essentially means that it can analyze a wide range of particle size. Okay, and the way the detectors are arranged when you have multiple detectors is shown in this diagram over here, where you can see a detector at 15-degree angle, which is to capture the forward scattering. And apart from that, you can also see two more detectors, one at 175-degree angle, which is basically to capture the backscattered signals which come out from this front wall of the sample cell. And there is one more at 90-degree angle, which is to capture the scattered signals from the side walls. Okay. And now which detector to be used and how the signal will be captured, that will depend on what kind of sample you have. For example, when the detector is kept at this location at 15-degree, that is primarily for capturing the forward scattering, and this kind of forward scattering happens for samples which will typically have smaller particles with few large particles which scatter in the forward direction. And this detector at 15-degree is also used to monitor aggregation in the sample, right? And the other two detectors are there basically to capture the backscattered signal. This one is to capture the backscattered signal from the front wall of the optical cell, as I said before, and the one at 90-degree is used to capture the backscatter signals from the side walls. Okay, so which one to be used to capture the backscatter signal will again depend on the type of sample. For example, this 175-degree detector is used for concentrated turbid samples where the particle concentration is high, and this 90-degree detector is useful for weakly scattering sample of small particles, right? So with the help of these multiple detectors, the instrument allows a range of sample types to be analyzed, and therefore it allows more flexibility to the system. Now, when you talk about backscattering, the path length of the scattered light can be reduced because the scattering volume over here is close to this front wall of the sample cell, and therefore the path that the light has to travel to reach the detector is reduced, and this will in turn reduce the multiple scattering which might interfere with the actual signal. Multiple scattering is nothing but scattering of the light beam by more than one particle before it reaches the detector, and if that happens, it might interfere with the signal, as I said, and the data may be skewed compared to the data which would have been received through single scattering. Now, in order to control this light path, focusing lenses can be used, with the help of which the focus point can be varied, as you can see from here. And as the path length is reduced, the multiple scattering is also reduced, and depending on the sample type, this focus length can also be increased if there is a need. For example, if there is a dilute sample of small particles, there may be a need to have more scattering from each of these particles, and therefore it may be beneficial to increase the path length, and therefore the focus point can be changed to a larger distance. On the other hand, if you have large particles or samples with high concentration, in those cases it will be good to have the focus point closer to the front wall of the sample cell to minimize this path length, and in that case the position of the focus point can be brought closer to the front wall of the cell. So that was all about the DLS instrument. But there is one more thing which is very important in case of DLS measurements, and that is the sample preparation. So having said that, one thing I must mention over here is that having a good suspension is key to the success of this technique, because if the particles are not uniformly dispersed in a liquid and if they agglomerate, then obviously the particle size data which is received is not a true representative of the actual particle size, and also detection of agglomerate is difficult by this technique, and therefore it is essential that a good suspension is made before the measurement is carried out. So these are the four vital things that one has to keep in mind in order to make a good suspension, and these are as follows. Selection of an appropriate liquid in which the particles are dispersed is very important, because every particle may not disperse in every liquid, and therefore depending on the type of particles, a particular type of solvent has to be chosen. Now once the liquid is chosen and the particles are added to the liquid to make the suspension, it is essential that the powder particle surface is weighted by the liquid. That means the particle surface should be entirely covered by the liquid. Otherwise, a good suspension will not form. Okay. So weighting of the particle surface is also very important, and this is something that we need to discuss in little more detail, and I'll come to that in a little while. And then, as I said, agglomerations are not good for this measurement, and therefore the particles need to be deagglomerated once they are added to the liquid. And in fact, a stabilizing agent is added to make sure that the particles do not agglomerate, and a uniform suspension is obtained where the individual particles are dispersed without forming clusters. So talking about the solvent which is used to disperse the liquid, water is the most preferred solvent for dispersing most of the particles unless they are soluble in water, and many times a low concentration salt solution is used instead of pure deionized water. That is because DI water can reduce the charge on the surface of the particles, and as a result the repulsion between the particles will decrease, and that would lead to agglomeration, right? Addition of a trace amount of an ionic compound such as sodium chloride can overcome that problem. For example, a 10 millimole solution of sodium chloride can be used instead of pure DI water to make sure that the particles do not form agglomerates. Other solvents can be also used apart from water, and the next best solvent is alcohol. For example, isopropyl alcohol, methanol, ethanol, all these can be used to disperse particles. Now this is very important. As I said before, the particles have to be weighted by the liquid Otherwise, they will not be suspended and a good dispersion cannot be obtained. Okay. So in order to do that, you will have to make sure that the particle surface is conducive towards water. That means it should not be hydrophobic and expel water. Rather, it should be water loving or hydrophilic so that the water can spread over the surface and coat it uniformly and wet it, right? So in order to do that, many times a compound known as a surfactant is used. Surfactant, if you expand this term, it stands for surface active agents, and these kind of molecules have two ends. One end is polar or hydrophilic, and the other end is non-polar or hydrophobic. Okay. For example, Triton X 100. Here you can see these two ends which are colored in different colors. The red end is basically the polar hydrophilic end, and the hydrocarbon chain that you see on the other end is the non-polar hydrophobic part. Okay. So basically, if you see it from that perspective, it has a hydrophilic head or the polar head and a non-polar or hydrophobic tail. Okay, like this. And how does it help in wetting the particles by the liquid? Now, when you add this surfactant in small concentrations, these surfactant molecules can get attached to the particle surface like this. And now that this surfactant is present over the surface of the particles, the water can easily get attached to the particle surface with the help of these hydrophilic heads which are extended towards the water, right? So this is how the surfactant will help in wetting the particles with the liquid in which they are added. Now, one thing you must remember here is the concentration of the surfactant that is to be used, because this is a very key parameter to ensure that there is no overdose of the surfactant, because if that happens, then there is a problem. But before we come to that, let me also tell you that there are different types of surfactant. Depending on the nature of the polar head, there are primarily 4 types. These are as follows. Non-ionic, where there is no charge in the polar head. Anionic, where the polar head has negative charge. Cationic, where it has positive charge at the polar head. And zwitterionic or amphoteric, which can have both positive and negative charge depending on the pH of the solution. Okay. So here are some examples of different types of surfactants. Triton X 100 that we saw in the previous slide is a non-ionic surfactant, and in the other categories you have this kind of compounds. For example, sodium dodecyl sulfate or SDS is anionic. Similarly, this compound, which goes by the trade name Adogen 464, is cationic. Similarly, this one CaHS is zwitterionic. Now, I was mentioning about the importance of the concentration that you need to maintain when the surfactants are added. They should be always added in low concentrations. For example, a typical concentration could be 1 ml per liter of the solution. So what is going to happen if you have high concentration? That can be understood from this particular image over here, where you can see a lot of these surfactant molecules coming together and forming an aggregate like this. Okay, so if the concentration is high, instead of attaching to the particle surface, these surfactant molecules would rather attach to each other and form an aggregate like this, and these kind of aggregates are known as micelles, okay, in which the hydrophobic tail of the surfactant molecules orient towards the center and these heads are oriented outward. So this kind of aggregates will form in the solution, and this is not good for the particles as far as their weighting is concerned. Okay, so therefore the concentration should be always low when surfactant is added, and there is a critical concentration beyond which these micelles form, and that particular concentration is known as the critical micelle concentration or CMC. So CMC is the concentration of surfactant above which the micelles will form, and therefore when the surfactants are added in the DLS suspension, the concentration should be always below the critical micelle concentration. So this is something that you need to keep in mind when you are preparing DLS suspension using surfactants. Now, once added into the solution, the particles would tend to form aggregates because of the attractive van der Waals forces between them. And therefore, the next important thing in making DLS suspension is to ensure that the particles are suspended without forming agglomerates to be able to scatter effectively, right? So in order to break these agglomerates, energy has to be applied. For example, the suspension can be subjected to ultrasonication for a particular period of time with a certain amount of energy, which will ensure that the agglomerates, if there are any, will break and the particles will be suspended in the liquid individually. And many modern instruments, in fact, are equipped with inline ultrasonicators to do the job. And when you do this ultrasonication, the applied energy has to be optimized. And these kind of instruments can even do that as well. So now we are done with three important things out of the four that we listed in the beginning. That is selection of an appropriate solvent, then weighting of the powder particles by the solvent, and then deagglomeration of the particles in the suspension. Now, even if you do this deagglomeration by ultrasonication, there are chances that the particles will agglomerate again because the van der Waals forces are always there to attract the particles to each other for them to form aggregates, right? So in order to avoid that, what is done is a compound is added to the suspension to stabilize the dispersion and prevent reagglomeration of the particles. Okay, so therefore this kind of compounds are known as stabilizer. Stabilizers are actually compounds which can create charge on the particle surface, and due to this increased charge on the surface, the particles will now repel each other, and this repulsive force can now overcome the van der Waals forces which are responsible for forming the agglomerates, and as a result of that, the agglomeration of the particles will be prevented. Okay. Sodium hexametaphosphate, or SMP, is a very commonly used stabilizer for making DLS suspension. And the stabilizer is also added in small concentrations. For example, 0.5 gram in a liter could be a typical concentration. Right, so these are the four important things that have to be followed in order to make a good suspension which can be used for DLS experiments to do particle size analysis. All right, let us take a moment to summarize this lecture. In this lecture, we have learned about the DLS instrument, which basically consists of a laser as a monochromatic light source, and then there are detectors, photodiode array detectors, which are used to capture the scattered light beam intensity, and then the sample, which is a fluid containing the dispersed particles, is sent through the sample cell, which is placed in between the laser and the detector. And as the particles come in the path of the light, it gets scattered, and the scattered intensity is recorded on the detectors and are sent to the computer to analyze the data using the autocorrelation function, and from that the particle size is derived. And we have also seen that there can be multiple detectors in the same instrument, and this will increase the dynamic ratio, which is the ratio of the largest particle size to the smallest particle size, and will provide more flexibility to the instrument in terms of handling different types of samples. And we have also seen that the focus position from where the data is collected, or the scattering volume, can be adjusted with the help of focusing lenses, and this can also be used to reduce the light path and control multiple scattering if there is a sample where there are chances of multiple scattering. And one thing that we have learned today is that it is very important to make a good suspension, because this is a key to the success of this particular technique. If there is agglomeration or clustering of particles in the suspension, then the accuracy of the results will not be good, and therefore it is always recommended that these things are followed. These four things which are listed here are followed to ensure that a good suspension is made for doing the DLS measurements. And these are selection of a particular liquid which is good for suspending the particles, weighting of the particle surface by the liquid which is used for dispersing them, and to make sure that there is no agglomeration of the particles, so there should be certain provisions to deagglomerate the particles. And finally, the suspension has to be stabilized to make sure that the particles do not reagglomerate again and form those agglomerates or aggregates which are previously broken by applying energy while deagglomeration was done. Okay, so these are the four very important things that one has to follow in order to ensure that a good suspension is made. Now, as far as the solvent or the liquid is concerned, water is the most commonly used solvent because it can dispersed a wide range of particles as long as the particles do not dissolve in water. And apart from that, apart from water, dilute salt solutions and alcohols are also used. And at times, in fact, salt solutions are better than pure DI water in order to prevent agglomeration of particles. And alcohols like IPA, ethanol, methanol, etc. can be used as the solvent for dispersing a variety of particles. And we have also seen that surfactants are used to improve the weighting of the particles in the dispersing liquid. Surfactants are actually surface active agents which help in weighting the particles with the help of the two ends that they have, the hydrophilic and the hydrophobic ends that the surfactant consists of. And when the surfactants are added, you have to ensure that they are added in low concentrations, because if added in higher concentration, they can form aggregate known as micelles. And there is a particular concentration beyond which these micelles form, and this is known as the critical micelle concentration, or CMC. And therefore, the surfactant which is added in DLS suspension has to be below the CMC concentration. And in order to make sure that there are no agglomerates, energy is applied to break the agglomerates, and most often this is done with the help of sonication. And we have seen that most of the modern instruments nowadays are equipped with inline sonicators which help in breaking the agglomerates. And this equipment are also capable of optimizing the energy that is to be applied, because when you try and break the agglomerates with the help of applying this kind of energy like sonication, it is also important to optimize the energy that has to be applied to a particular type of sample. So with that, we come to the end of this class. Thank you for your attention.